start. Good morning, everyone. Today I'm going to talk about data science and analytics in aviation. With the development of advanced technologies, currently a large amount of real-time data are all available from a diverse set of sources. This paper focus data science in analytics in aviation studies in several critical areas, such as big data analysis and air transport network management and machine learning. So, in this slide, they have stimulated new research ideas, knowledge techniques, and application in addressing new problems. Furthermore, with the development of big data analytics, as well as blockchain, a new area of aviation research has begun. For example, flight data regarding delays was studied and find that delays are highly related to flight departure delays. Data science and data analytics has been a popular issue in the last decade, especially to aviation study. Uh, for, for example, here, Table 1 provides information about the numbers of publication in the last decade. We can see that there are very few publications in the period of 2009 2012. From 2015 through 2019, there is a rapid and accelerating growth. And in a vision study, forecasting, machine learning, air transport networks are critical topics. Let's see four principal areas. Regarding big data, in the last year, researchers in different disciplines have realized the benefits of big data. This can provide new knowledge to researchers and professionals. At the same time, big data analysis it's a hard topic, as many of these data sets are high dimensional and insufficient. That can be easily manipulated by classical static methods. Recent advances in applying mathematics have introduced new topological methods which offer a new direction from which the salient highlight features big data may be obtained. These methods have already found a wide range of data science application, from network neuroscience to path a planning for autonomous robotic system, as well as aviation data analytics. About forecasting, about forecasting in aviation, we can say that the flight delay prediction is one of the most crucial topics. Forecasting with big data is one of the latest prevalent application in areas such as airport and flight delays, promotion planning, etc. With high dimensional data, a novel deep network model is developed to mine the inner deep pattern of flight delays. The proposed model has been proven to be highly capable of handling the challenges of large data sizes and capture the key factors affecting delays. Regarding machine learning, uh, regional convecting weather, it's analyzed how regional convective weather affects the ground delay program. Um, uh, by applying support vector machine logistic regression and random forest based methods, it's discovered that using a properly tuned support vector machine can identify the importance of convective weather. This information is very useful for flight operators. About air logistic, it's an important to say that the routing problems are classic in here. To do so, they analyze a set of data collected using neural network approach to predict the relationship between the price and demand for maintenance providers. They develop a Stockbeller Nash gaming model, which considered the price competition among maintenance providers. The results reveal significant saving for the airlines and maintenance providers.
1 minute
And,
left.
finally, okay, and finally, we can say several important future research directions are proposed, such as blockchains. It can be used to enhance the protection of privacy information like personal data, data quality in the aeronautical industry. Data security is also very important. For example, airline record billions of passenger personal information. On the other hand, Industry 4.0 is mainly about applications technologies originally in the manufacturing sectors. With the application of automation technologies, researchers are now studying how to replace the physical air traffic control tower with a virtual one, the success of which is believed to bring saving and operation expenses. So that's all. Thank you for your attention.
Good morning. Can you hear me? Yeah. Okay. Okay. Good morning, everyone. In this occasion, I will talk about this topic. It is microfluidic device with brain extracellular matrix promote structural and functional maturation of human brain organoids. But the idea of this article is develop a methodology and also they use a microfluidic device in order to prove how the human organoids develop in a microfluidic device, but with this BEM. They call it BEM, but is a human brain tissue that is derived from extracellular matrix. It means they use this tissue from patients they include in the diet to differentiate human pluripotent cells from brain, and they compare this with this normal tissue that they call it macrosoma proteins without this BEM. Uh, this paper is related and they characterize what is the difference between introduce a tissue extracellular brain from patients and without this tissue. First, this is the scheme that they use for along the days to develop the organoids. In the 50th week, they introduce this tissue from brain, and then they characterize what is the difference between them, no? Between the mat that they call and the BEM. And they found that the principal difference that they found that the BEM are very similar for human cells brain, and also the mat they have a lot of glycoproteins, but they tested in different batch in order to see what is the reproducibility of this BEM, and they tried three different batches and they found the similar there are not too much difference from this batch, And they also found a different kind of proteins that are more related with human brain in BEM organoids. This paper is huge. I will talk a little faster, but I will try to explain very quickly because there are a lot of proofs that I need to explain. And the second is that they found that the BEM is a huge organoid, is more than MAT. The larger of this is greater after the 30 day, and also the protein is that is basal lectin are they found that there are more in BEM in along of the days. Yeah? What is so important is this because they use these markers in order to see what is the difference. But the main difference is that there are more along of the days, and also there are more, and also they are more stable. It means that they don't lose along of the day. That this is so important because this basal membrane is important for develop a 3D structure, and also the radial structure of this. And also, when they fold this, they say that it's also important for to construct neurons, a good health of neurons. That is the reason they saw this. And this is, I told you, the 3D structure of Ben is more, the construction of the 3D structure is better than math. Okay. Another test that they are interested in is looking for the genetic expression. They made this Pearson correlation, and they found these MAT and BEM are very close to the first days of the fetal organoids. It means that the cells are very similar from the brain of the fetal and the first weeks. You know? The more of the correlation of both are in the same. The expression genetic is in this area. Okay. Okay. After that, they asked why BEM is so important, because they introduced this tissue from brain, and they also use a different controls. The one is with using intestine, brains, liver tissue, and hepatic also liver and heart, sorry, heart tissue, in order to see if they also increase the size of the organoids, introducing different type of tissue. But and again, the brain tissue also develop a longest organoids, and also they found the expression also of these genes are also more involved in BEM. Okay, this is the microfluidic device. I have another in support material. This is the microfluidic device that they use. It's this bigger chamber, and then in the middle they cultivate the cells, and they develop the organoids in these channels. They don't use a pumps. They use this
1 minute left.
this machine in order to move the medium for this direction. And this is the another question that they try to find: what is the interchain of glucose, if the cells are consumed as well, good. And they found that in the device is better than use a normal plate. And also there is the difference when you use the normal plate, the BEM plate, and the BEM device. You know, this is when you use a microfluidic device. Also, the organoids are bigger, and also the develop of this radial construction of this is better. And also they measure the current in this, because they are also interested to characterize what is the response of the cells under the current, and they then found a similar behavior with the BEM plate and the base plate. And finally, To end, in order to see the reproducibility, I talked about that they use different batch and they don't find too much difference with these batch experiments. Thank you.
Good morning, everyone. Today, the topic I'm going to talk about is a microfluidic system for applying shear flow to endothelial cells on culture insert with collagen Vitrigel membrane. So here, the authors they propose a microfluidic system which is built. It comprises an insert here in skin A from figure 1. You can see this insert where this membrane is assembled, and at the bottom of the membrane there are a culture myotubes, and in the other side they culture endothelial cells. So it's important to study the relationship between cells from the muscle and how endothelial cells interact to understand how insulin reaches the muscle cells. So here on the membrane, they functionalize it with this collagen Vitrigel membrane, and they place this insert inside these reservoir, and the flow, as you can see, is going to apply a shear stress on the endothelial cells. So here there is a zoom in where you can see the membrane, the myotubes, and the endothelial cells in yellow, and the nuclei are stained with DAPI. So they are going to measure from the images, they are going to measure the orientation of these nuclei and how the cells align in the same direction of the flow. So here is a schematic of the microfluidic system. You can see that there is a tank, and it's like a bubble trapping mechanism. So to avoid that the bubbles enter to the tubing that connects to the microfluidic system, they use a peristaltic pump, and this peristaltic pump also pulsates, but they address this issue by using this fluidic tank, which decreases the pulsation from the peristaltic pump. Okay, so because they want to measure the shear stress that they cause to the cells, they use this formula here to measure the shear stress, where they perform a model with console in order to measure the speed, the average speed. As you can see here, the average speed and the height of the microfluidic channel are represented, and also they enter to the program the viscosity, which is here. Okay, they perform this flow simulation, and after they characterize the system, they also use this statistical method so that they measure the angle that the cells of the nuclei, how the angle, and they measure this variation, and then in order to see if the membrane is the endothelial membrane, they measure to measure its barrier properties. They use an electrode, and they measure the electrical resistance. So here is the formula for the electrical resistance. And here we have a side view of the channel, and also the top view of the channel, how the speed varies, as you can see here in the color map. They measure the transendothelial electrical resistance, and also you can see here how the cells look when they use the tank, or when they don't use the tank. You can see how the cells, the nuclei alineates to the flow, and also we see here the variation of the angle of the cells. So they have proven that they can apply and control the shear stress according to the speed of the flow, and they measure the angle that the cells are orientated according to this shear stress. Here, they also marked the myocytes and the endothelial cells, and they thank you. Yep, and they conclude that with they can culture these two types of cells with perfusion, and they can solve these problems of the pulsating with the pump. Okay, thank you very much.
Okay. Good morning, everyone. Today I will talking about of this article called an automated method for the analysis of food intake behavior in C elegans. Well, here they present an automated bioluminescence-based method for the analysis and continuous monitoring of worms feeding in a multi-well format. They validate this method using genetic, environmental, and pharmacological modulators of pharyngeal pumping. This study is important because the rate of food intake determines the rate of growth and anomalous feeding behaviors that are associated with an assortment of chronic diseases such as obesity, type 2 diabetes, cardiovascular diseases, and some cancers. They use C. elegans because the metabolism and central signaling pathways are highly conserved between mammals and these organisms. And the regulations of feeding is not an exception. C. elegans feeding occurs as a consequence of two motions. The first, the cyclic contractions of the pharynx or pumping, and the isthmus peristalsis. And the feeding rate is proportional to the warm pharyngeal pumping, where bacteria are sucked the environment and grinded up. This study is a new approach that is based on a method where they measure bioluminescence in strains transgenic that express luciferase. The luciferase catalyzes the conversions of the substrate luciferin into oxyluciferin in a reaction that emits light. They measure the luminescence from a single animals that express the luciferase fuse to GFP in the L4 larva, the worms, and that ingest luciferin and emit light until they reach for the 4 molt. Okay, here in this figure, you can see the luminescence signal reports food intake of single animals. Here you can see the bioluminescence signal for single transgenic animals. The absence of substrate intake provokes a decrease in the bioluminescence signal that resumes when animals exit the fourth molt and enter the young adult stage. Here they present a pharyngeal pumping per minute of wild type and these do mutant animals transgenic, and the results in strains have reduced pharyngeal pumping rates. Here you can see the mean luminescence signal emitted from the mutant worms was reduced compared to wild type. And here present the total GFP fluorescence intensity. Among the strains, don't exist significant difference in the fluorescence. They realize assays with a small group of worms and compare the wild type and this mutant strain. The signal exhibit interruptions and reductions in the luminescence signal compared to the wild type. Okay, here realized the luminescence sign response for the for change in food availability and quality. Normally, for in laboratory conditions, C. elegans is fed on E. coli strain OP50. But in this study, we measure differential strains. This you can see here, and compare to strain OP50. Okay. Here, you can see in this figure the mean bioluminescence signal for single transgenic animals at adult young stage. And the worms were fed with these strains, and the luminescence signal emitted by worms feeding on was significantly lower than the one of on OP50. Here you can see the description of the experiment of fasting and refeeding. The animals also reduce their pumping rate when deprived of food and increase it again when they encounter food. They measure the response of C. elegans to change in food availability using luminometry. And here you can see the mean luminescence signal of 2 hours measurement for each conditions when there is only is basal in the well. The animals show reduced luminescence signal compared to the animal transferred to wells with OP50. Okay. And in this part, okay. They use to these drugs, serotonin, naloxone. Serotonin is increase pharyngeal pumping frequency of young adult animals, and the naloxone is a morphine antagonist that inhibits food intake in starved worms. In this figure, you can see the mean pharyngeal pumping per minute of single transgenic animals in the absence and presence of serotonin. The drugs treatment increased pharyngeal pumping frequency of young adult animals. Here you can see the mean luminescence signal in the absence or presence of the same drugs. And the luminescence signal was higher in the presence of serotonin as compared to the control conditions. Here they presented total fluorescence intensity and the mean pharyngeal pumping per minute in the absence of presence of naloxone confirmed that the drug treatment reduced pumping rate under these experimental conditions. And the mean luminescence signal from individual in the absence of presence of naloxone, and the luminescence signal was lower in the presence of naloxone, and here you can see the total fluorescence in the absence of presence of this drug naloxone. Did not find a significant difference. In conclusion, the method described here adds an invaluable tool to investigate how internal and external cues modulate feeding behavior. Okay, that is all. Thank you.
Hi everyone, I'm going to be presenting this paper. And here, the authors, in order to better understand the impact of heterogeneity and complexity of tumor microenvironment, they developed a microfluidic-based platform for real-time monitoring of drug delivery that approximates in vivo tumor barrier characteristic to reproduce the enhanced permeability and retention effects, the APR, with permeability values that are similar to those reported in vivo. So they use a soft lithography-based process to construct this chip that has both vascular and tumor compartments, separated by a tightly packed cylindrical micropillar array. The geometry of the vascular channels and dimensions closely approximate the size and morphology of microvessels in vivo, and they permit the platform to maintain physiologically relevant shear flow conditions in the vascular channels for endothelial cell growth. The tumor and vascular compartments are separated by a micropillar array interface that allows direct communications of the tumor and the endothelial cells. Co-culturing of the tumor cells and the endothelial cells was achieved by first establishing the endothelial cells in the vascular compartment, and then followed by the culture of the tumor cells in the tumor compartment. Human breast cancer-associated endothelial cells were cultured on the fibronectin gelatin-coated inner surface, and they form a continuous 3D lumen on the vascular channels. The permeation of fluorescent dextran from the vascular compartment to the tumor compartment, either with and without the presence of tumor cells, was characterized by measuring fluorescence of the dextran. Culturing the endothelial cells in the vascular compartment of the chip significantly decreased its permeability to the fluorescent dextran compared to the chip with no tumor cells. The permeability of the endothelial cells in co-culture with tumor cells was measured to determine how the presence of tumor cells impact the enhanced permeability and retention effect. Changes in permeability of the endothelial cells in co-culture with tumor cells are consistent with morphological changes in the endothelial cells. With prolonged co-culture, the endothelial permeability increased significantly. The effect of co-culture on permeability and the endothelial cells morphology was more prominent in highly metastatic MDA-MB231 tumor cells as compared to less metastatic MCF7 tumor cells. Also, co-culturing either metastatic or non-metastatic tumor cells with the endothelial cells significantly increased liposome extravasation as compared to culture without tumor cells. So the presence of metastatic MDA-MB231 tumor cells changed the endothelial cells phenotype and increased its permeability. Highly metastatic tumors are known to be able to disrupt endothelial junctions and therefore increase vessel permeability, and B-coloring is required for forming tight vascular barriers, and its downregulation induces endothelial barrier leak. When cultured without tumor cells, the endothelial cells form continuous intercellular adherent junctions under shear flow. But in the presence of tumor cells, the formation of adherent junctions between endothelial cells were reduced. So, metastatic cells clearly downregulated adherent junction expression, as indicated by the disappearance of big gathering in some cells, as well as intermittent big gathering expression along the edges of the remaining cells. Finally, the endothelial cells tight junction molecule expression without tumor cells or in co-culture was identified by immunostaining. When cultured without tumor cells, the endothelial cells exhibit homogeneously distributed molecule expression, and with the presence of metastatic tumor cells, it was only detectable at a few spots between contracting endothelial cells. So, in agreement with in vivo observations, these results indicate that highly metastatic MDA and B231 tumor cells are capable of regulated big gathering and tight junction expressions in endothelial cells. Altogether, these results demonstrate that the presence of metastatic tumor cells modulates cell-to-cell communication as well as endothelial cell barrier function, which has significant implications for developing targeted drug delivery strategies. Thank you.
Okay, this is my topic: identification of attractive odorants released by preferred bacterial food found in the natural habitats of C. elegans. Okay. The choice of food is essential for the survival of microorganisms. One should choose nutrition foods in this paper. Tests on food other preference for C. elegans in four out of 6 bacterial isolates, isomyl alcohol was released. A well-studied C. elegans detects isomyl alcohol at low amounts. This odor is relevant, released by some bacterial isolates in the environment of C. elegans. In this part, in part A, bacterial isolates were evaluating the environment of C. elegans. The strains were isolated for Lampest, rotten apple, slug. In this trail, C. elegans show a significant preference over E. coli HB101, Providence, Alkaligenesis, Providencia, Pseudomonas, and moderate preference, Rotella, no preference, Actinobacter, Flavobacterium, Pseudomonas sp. Okay. In part B, C. elegans did not show a significant preference over JUV T9. In part C, the growth rate of C. elegans in this bacterial isolate was evaluated in relation to the growth rate of E. coli OP50. And part D, in general, the bacterial species were JUV4, 5, and 39. And the section was the preference in Pseudomonas JUV12, but it didn't improve growth. And in this part, is volatile organic compounds or VOC are present in bacterial isolates. A mass spectrum was used. Chromatographic peaks, blast abundant are greater than 6 per 10 7. Low abundant was between in this part between 1 and 6 per 7, and figure 3, 5 of the 6 attractive natural isolates had 2 or 3 dominant high abundance compounds are several low abundant compounds. Among them, isoamyl alcohol, 2-methyl 1-butanol, dimethylisulfide, ethylisobutyrate, and ethyl isovalerate. These compounds are attractive in this study, and this part, 2-methyl butanol is attractive to C. elegans over a range of concentration. Methylisulfide is weakly attractive. In addition, there are several others that are attracted to worms, such as 2-methylpropanol, acetone, 2-butanol, isomyl acetate, and ethyl acetate. And conclusion, the results can ensure that C. elegans prefers bacteria that improve growth rate, but many will need to be tested on more isolates to support this conclusion. Thanks.
Can you see my screen?
Yes.
Yes.
Thank you. Good morning, everyone. The paper that I will talk about today is single cells tracking reveals super spreading brain cancer cells with high persistence. This paper studies the trajectory and velocity in cancer cells using machine learning. They find that the speed of displacement has preferential direction of motion against the hypothesis of random directions of movement. The cells studied are from brain glioblastoma tumors. The experimental conditions are the cells cultures obtained form a spheroid of 278 micrometers of diameter, an aspect ratio of 1.32, with a density, of 30,000 cells per cubic micrometer. To quick starvation micrometer interchange growth medium with the ECM formulation Vitrigel, which consists of laminin, collagen, fibronectin, and various growth factors, and that mimics basement history membrane. We can see in figure 1 invasion migration of cells right after the transfer to Vitrigel at different times. times 0, 8 hours, 18 hours, and 1 day. And we can see the growth of the spheroid. Spheroid because the invasion cells occur in these systems. In figure B, they obtained the time-dependent average invaded cross-sectional area for 25%, 50, 77, 67, excuse me, 75 and 0%. These percentages correspond to the concentration of Matrix. And they confirm that the cross-sectional area of a tumor spheroid grows linearly with time in fresh cultured medium. In contrast, for the invading spheroid, they found an exponential growth. For example, the blue line. Figure 1D shows the population of cells with a different cross-section obtained. The bar corresponds to the error calculated. In figures 2A show some example of migration cells trajectory. The colors correspond to the time and the length of the line correspond to the pace of motion of the cells. The scale corresponds to sin 100 micrometers per centimeter. Figure 2B corresponds to the distribution of displacement of a step cell trajectory. Cell trajectories are highly diffusive, but in contrast to the invading cell collection, the migrating cells seem indifferent to material density. 25, 50, 67, and 75, the four of the distribution is equal. In conclusion, the authors say that it's indifferent to the concentration in material. Figure 2C shows the ensemble average means squared displacement, MSD. And they can see an anomalous diffusion of the migration cells. The exponent alpha is different of 1 in all cases. Alpha corresponds to the exponent to the relaxation time in diffusion theory of physics. In normal diffusion, alpha is 1, but in this case, alpha is different of 1 for all cases. Here, we can see the error calculated in this value. Figure 3A show the box plot of instantaneous velocity. Red line corresponds to the average velocity. Figure 3B correspond average migration speed. That means is to average the trajectories calculated. And finally, in figure 4a, the author showed the distribution of angle of velocity and displacement motion for times. And we can see the preference angles of motion. In conclusion, the cancer cells has a preference direction of motion. His dynamic is very interesting for study, and the authors saying in authors saying in showed, excuse me, the author showed that the few works in this direction of research. Thank you very much.
Hello, everyone. You can see my screen?
Yes, we see it.
Okay. In this case, I talk about it's not common topic we talk in this space, but for me it's very interesting speak with the interesting topic that relates on the our life now. Okay, in this paper, focus the intermittent fasting methodology and treatment for the obesity and COVID. Okay, in this the 18 months we began the pandemic, the life change, the sedentary mode increase. This the consequence is very severe, but in around the world increase the problem relates on the more weight in person. Okay, and in this review show the more evidence how intermittent fasting help to lose weight and measure the hypoglycemic. Okay, but in this case compare the healthy lifestyle approach. Okay, and show the more advantage in the different strategy for reduce the weight loss and the different metabolic healthy problem and the ends. Okay, and you can see the TRF on the intermittent fasting and reduce is the weight, the improved glucose homeostasis and sometimes the financial cost is more cheap and don't require reduce the energy needed to take but it's complete strategy and treatment for this problematic problem healthy. Okay, um, in this diagram how the obesity and diabetes is related when weight increase and the sedentary mode life is related all characterized for the several COVID complication health and death because in increase at the different proteins and how the thunder cytokines. Okay, in this part, you can see the different reaction for our body and the impact on the metabolic and in immune function low for obesity sedentary life related on the characteristics for more adiposity and inflammation and immunity, and so more in the case on the height weight in the person, the glucose and homeostasis, and increase the level insulin and resistant insulin, but is a consequence for increase the resistance of eliminate the toxin and the other problem, but inflammatory problem that you can see that is very difficult. And intermittent fasting is a great method for reduce in a low time this problem, how you reduce the hours for eating in the day, and the for example only eat in the 8 hours of the day, and the rest of the day I not not eat only drink water or different liquid, and no eat. Oh, in this case you can see leptin pathology reduce is when the people try the intermittent fasting. Okay, for me it's very interesting understand how our body respond, and the different stimuli is very interesting for me. I hope for you those things.
Hi everyone. Everyone can see my presentation.
Yes.
Okay, thank you. So here I'm going to show you this article called the Microfluidic Bioreactor for a New Era of Bioprocess Development. So this is about a system that use microteether plates with an integrated microfluidic chip. And they proved that this system can successfully carry out a small-scale fed batch cultivations with volumes between zero point 0.8 to 1.5 milliliters. So first, we have the research based on parallel fermentation. Here, the high throughput fermentation in microliter scale is the method of choice as it generates high data output while cutting costs due to savings on time and workforce, automation, and the low material input. The system that they use was the Biolector Pro has as a new tool for efficient bioprocessing. And this system is a fiber optic online measuring system capable of monitoring biomass, fluorescence, pH, and DO in continuously shaken micro teeter plates. This system is based on 48 well micro teeter plates. Microfluidic process control enables pH regulation and feeding in up to 32 parallel wells, individually operate bioreactors. And the remaining 16 wells of the cultivation plate are used as reservoirs for the feeding or pH control regions. The specific well's flower shape was developed in consideration of decreased spilling and foaming of the fermentation broth. I think this is interesting that we can use. And to guarantee the sterile condition, the micro teeter plates is sealed with a sterile membrane that includes silicone septa for sampling. Here we can see in this figure how is how it looks this bioreactor and the samples here. And this is the culture wells, and we can see the points with the pH channels and the feeding channels. The principle, as the first two wells rows are used as reservoirs for the desired feeding solutions, the remaining 32 wells work as distinct bioreactors. The microfluidic technology utilizes pressured air to actuate membranes valves at the bottom of the microfluidic chip, and the liquids are pumped through the chip via microchannels directly into the wells. So the progress of the 32 parallel cultivation is monitored and controlled in real time. In the figure 3 demonstrates a fed batch cultivation of E. coli with different feeding rates, as we can see here. And for each cultivation well of the microfluidic flower plate, a specific feeding rate was set, and with a glucose solution of 1500 grams per liter, feeding rates from the 0.6 to 10 microliter per hour were realized. And the microscale results with this device had previously been proven to be scalable to fully control 1 and 20 liter stirred tank bioreactors. And the same growth behavior was compared with C. glutamicum cultures. And finally, the microfluidic technology brings full processes control and precise dosing to microscale. Those parameters can be optimized at an early stage. And the technology paves the way for much faster research and development and the application of more advanced industrial bioprocesses. And this chip implemented into the microfluidic flower plate relies high throughput cultivations in batch or fed batch mode, together with online monitoring and control of the most common fermentation parameters. Thank you, that's it.